Thank you so much for inviting me to give this presentation. And as you'll see, I want to make this talk a tribute to Dr. Peter Weygoldt. So first, I want to thank the Laboratorio de Biología Reproductiva y Evolución in Argentina for inviting me, for putting on this seminar series. It has been amazing. You have attracted some incredible scientists, and I'm so glad you're doing this, and I'm absolutely honored to be a part of it. So thank you all so much, and it's great to see you. Okay, so I wanted this talk to be a tribute to Peter Weygoldt because he has been such an incredibly influential person in my scientific career. So in this slide, I have him up on a pedestal because he has been up on a pedestal for me since I first learned of his work. And many of you know that he recently passed. And in March of 2018, I just wanted to highlight that there was actually a special issue in tribute to his life and his work that Gabriel Uhl organized and was the special editor for. And a lot of this information about his life, I actually got directly from that. So thank you, Dr. Uhl, for providing more information about the personal life of Peter Weygoldt. So Peter Weygoldt went to Albert Ludwig Universität and Christian Albrechts Universität from 1952 to 1958. And during that time, he was advised by Adolf Romain, who was, Dr. Weygoldt was advised by Adolf Romain, and he was an incredibly versatile biologist. He was very well known for his work on morphology and phylogeny, and he was very influential for Dr. Peter Weygoldt's research. In 1958, he received his PhD, and his PhD was actually on the embryonic development of an amphipod, of a crustacean. Okay, from 1960 to 65, he worked at Free Universität in Berlin, and this is when he did a lot of work on pseudoscorpions. From 65 to 67, he was at the Marine Lab in Beaufort, North Carolina, as well as Friday Harbor Laboratories. And again, here he was working a lot on the embryology of crustaceans and the biology of pseudoscorpions. In 1967, he was offered a position back in Germany as an associate professor at Albert Ludwig's University, where he was promoted to a full professor in 1978. And this is where he stayed for the rest of his career. He did a lot of traveling to Brazil, South America. He traveled all over the world to do research on Amblipigids, but he also did work on other organisms during that time as well. So Peter Weygoldt is most known for a variety of conceptual areas. He did a lot of research on development, just the general biology, reproductive biology, and phylogeny of a variety of different animal groups. So he worked on crustaceans, on neotropical frogs, on pseudoscorpions, and on amblipigids. And it is the amblipigids that I want to talk to you about today. So I did my master's degree in an arachnology lab and took arachnology in that course during my master's degree. So I was familiar with all of the arachnid orders, and I was familiar with amblipigids. And I knew their placement phylogenetically. They were relatively closely related to spiders, but I had only seen them preserved in vials in alcohol. And it wasn't, so at this time, I knew that they walked on 6 legs. So for those of you who aren't familiar, although everyone here probably is, amblipigids have 8 legs, but they only walk on 6. And their first pair of legs are these incredibly long, thin legs called antenniform legs. So I knew this about their morphology. I knew that they were mostly nocturnal, that they used these big spined pedipalps to catch prey at night, that they were generalist predators. But I didn't really understand how absolutely mesmerizing and fascinating amblipigids were until I saw my first one alive. So in 1997, I took a tropical ecology course down in Costa Rica through the Organization for Tropical Studies. And my very first night on the course, we went out at night I went with one of my friends from the same university, and I saw my first live Amblipigid. And it was sitting on the side of a tree trunk, and it was moving its antenniform legs around in just this very seemingly purposeful, deliberate, and incredibly graceful movement. And I absolutely fell in love with these animals. So during the rest of the course, I did multiple projects on Amblipigids whenever I could, wherever we were. I tried to do an Amblipigid project. And then when I got back, I was at the University of Arizona, and I started reading everything I could about Amblipigids. And at the time, there wasn't a whole lot, and a lot of it was in German. But I did find that there were some Amblipigids that lived in the Florida Keys. So over Christmas break, I drove from Arizona to Dearborn, Michigan, which is where my family was living at the time. I convinced my parents to get in their RV and drive me down to the Florida Keys. And I started looking for Amblipigids. And I finally found them in the National Key Deer Refuge on Big Pine Key. So this is what the habitat looks like on Big Pine Key. And basically you flip over these limestone rocks, and underneath the rocks, the Amblipigids will be kind of hanging upside down. So I finally found my first Amblipigid literally on New Year's Eve in 1997 and brought them back to the laboratory at the University of Arizona. So I had done my master's degree mostly focusing on wolf spiders and questions of sexual selection and communication and mate choice. And so when I was doing my reading of Amblipigids, I learned that male Amblipigids build spermatophores, and that these structures can be incredibly elaborate and intricate, and that they vary tremendously across different families and different species. And not surprisingly, all of this work was done by Peter Weygoldt. So I was already very familiar with his work, and I was just fascinated by spermatophores and by the possibility that there is male mate choice, that males are investing so much in these spermatophores that they might be exerting serious choice on who they mate with. So my plan was that I was going to work on male mate choice in Amblipigids for part of my PhD. The first problem was that I had no idea how to tell a male from a female Amblipigid. I searched the literature I could read. I couldn't find anything. I reached out to fellow arachnologists that I knew. Nobody could help me. And again, Peter Weygoldt came to the rescue in a paper that was written in German that one of my friends, who was a postdoc at the time at the University of Arizona, kindly translated for me. So she was skimming all of these German papers to try to find any information about sexual dimorphism and how to determine males from females. So I was finally able to determine males from females. I went back to the lab and was ready to do some spermatophore studies. I had brought rocks back with me from the Florida Keys, and I had an arena all set up. I put a male on top of the rock. I had a red light since these animals are nocturnal. I was doing this at night in the lab. I put a male on top of the rock, and he immediately ran underneath the rock. And so I wanted to be able to film this. I had a camera all set up, and I wanted to be able to see what was happening. So I thought that's not going to work if they're underneath the rock behaving. So I got out duct tape, I used plastic, all sorts of things to try to contain the animal to the top of the rock. And after an hour or more of trying, I gave up. But then I thought maybe I could use something like an island. And so I got a bigger container and I filled it with water so that only the top of the rock was above water. I put the male back on. I went to grab the female. And as the male walked over towards the edge of the rock, he was reaching out with his antenniform legs, reaching for the outer edge, and he fell into the water. And I went to grab forceps to save him, and he flipped under and ran underneath the rock under the water. So I did what any scientist would do, and I started a stopwatch to see how long he would remain underwater. And he stayed underwater for more than 35 minutes and was walking around the entire time. So the next morning, I came in and I set up containers of water and rocks all across the lab. Every Amblipigid I had, I chased them into the water, and I started a stopwatch to see how long they could stay underwater. And they were remaining underwater for more than 9 hours. So this led to my first real investigation into Amblipigids to try and figure out how on earth are they able to do this. And through a series of experiments where I deoxygenated the water and looked at behavior, I was able to find that they actually could get oxygen from the water. And they did this through special structures on their cuticle on their book lungs. So I did some scanning electron microscopy to look at the opening of the book lungs and found that they have these really high buttressed cuticles, and that acts as a plastron. So it traps a layer of air underneath, and that layer of air acts just like a physical gill. So these Amblipigids in the Florida Keys could actually breathe underwater. So all of this happened just before my first International Arachnology Congress, which was in Chicago at the Field Museum at 1998. And everything came together there, where the logo for the meeting was an Amblipigid. Dr. Peter Weygoldt was a keynote plenary speaker at that meeting, and I presented this research where Amblipigids could breathe underwater, and I won the student paper competition. And it was the first time I met Peter Weygoldt, and I was so nervous. I was, a new PhD student, and he was so kind and so encouraging, and that started a long relationship that I had with him following that. So also at that meeting, I met Dr. Rainer Felix, and many of you know him because he wrote The Biology of Spiders. But what you may or may not know is that Felix had also done a lot of work on Amblipigids and had published quite a bit on Amblipigids. And for those of you who may not be familiar with this older version, this is the newer version of Felix's work. So I met Felix at this meeting as well, spent time with him talking about Amblipigids, and really got interested in the sensory system of Amblipigids. So I went back to Arizona and started to look at the tip of the antenniform leg. So I got really interested in these legs. And doing some scanning electron microscopy, when you look at these legs, you see that they're covered with sensory hairs, and they are different sensory hairs that have different functions. So there are these big bristles right here that have a socketed base, and these are mechanosensory hairs. But at the tip, they also have a pore. So these are also contact chemosensory hairs. There are these little club-shaped sensilla, which have a pore at the tip that you can see over here. These are also contact chemosensory sensilla. But these were the hairs I was really intrigued by. Felix and others had suggested that these hairs had pores along the whole shaft, suggesting that they were actually olfactory hairs, that these animals could smell with these hairs. So one of my first electrophysiology studies focused explicitly on these hairs, and I was able to demonstrate that indeed Amblipigids can smell, and that they can smell just as well as most other insects. And this is not common for arachnids. But I really wanted to learn more about the animals themselves, how they're living in their natural environment. So I got funding to go back to Costa Rica to La Selva Biological Station, where I did a longish term field study, where I did a mark recapture study in plots that were 50 by 25 meters long. And I would go out to these plots every 2 or 3 nights and check for Amblipigids. Every individual was marked, so I knew the individuals, and I knew what tree they were on and where they moved. So what I found from this study is that Amblipigids have really high site fidelity. They can travel really far. They can travel more than 30 meters. They often return to the same tree. And just based on movement patterns, there's a suggestion that they might be territorial. But this just led to more questions. So how are they using their antenniform legs in this environment, and how do they find their way? And while I had been doing this field work, I had made some observations in the field of behavior and noted that when two individuals come together, they often fight, and that the fighting involves antenniform legs. This was also right around the time that Peter Weygoldt's book came out. So it was published in 2000. The title is Whip Spiders, Their Biology, Morphology, and Systematics. So this book had not been available to me when I first started, but as soon as it came out, I read it cover to cover, have read it many times, carried it around with me everywhere on the off chance I would run into Peter Weygoldt. And sure enough, at a meeting later, I was able to get my book signed. And I still have it with me here with the signature. And it is one of my prized possessions. So again, I was really interested in this antenniform leg and in how these animals use them, and had noted that they might use them in contests. So in collaboration with Casey Fowler-Finn, who at the time was an undergrad at Cornell University, we did a study looking at contests in Amblipigids. So I'm going to show you, this is real time with a night shot camera. So this is taken in complete darkness, two Amblipigids fighting. So you can see that they do this fencing behavior with their antenniform legs. This is a side view. And then if that doesn't determine the outcome, it can escalate into an actual battle. And so this is a battle looking at it from the top, and then we'll see a side view in a minute. Okay. So what we found out in that study is that they do use their antenniform legs in this behavior that we call antenniform leg vibrations, and that whoever does that longer wins the contest. So several years later, Dr. Roger Santer was a postdoc in my lab, and we wanted to explore the use of these legs in contests a little bit more. So the first thing that we did is characterized what proportion of contests have these antenniform leg vibrations. And it turns out that 74% of contests, animals will do this display. Using high-speed video, we showed that these legs actually do not come in contact with the body. So it's not a physical mechanosensory signal that individuals are sending to each other. So we hypothesized, well, we were trying to figure out how they were communicating with these legs. So Roger went back to all of our videos and looked at where over the opponent's body the leg most often was positioned. And what you can see here is the darker areas are areas where an opponent positioned its antenniform leg. And when you look at the patella joints of these walking legs, what you see is that there are two long, thin sensory hairs that are trichobothria. And these hairs are sensitive to airborne movement. So we hypothesized that these legs are being waved over these trichobothria, and that it's actually an airborne signal that is being sent in these contests. So in order to test that, we restrained an Amblipigid. This is mostly by we, I mean Roger, designed a motor where we had a wire that was the same length and thickness as an antenniform leg. It was hooked up to a motor that would move at the same rate that these animals would wave their legs. So it would move over these hairs, and then he put an electrode in and recorded the response. And it turns out that every time this wire passed over those hairs, we got an action potential. So these animals can detect air particle displacement, and it turns out that they're using this during contests. So in a follow-up study, we actually clipped the hairs. And if you clip those hairs, then the duration of antenniform leg waving no longer predicts who wins. What's really neat is that there's a close match between the rate at which the animals signal and the capacity of those hairs to detect signals. Oh, and this was the first time these hairs had been shown to be used in animal communication, in any animal. Okay, so here on the x-axis, we have the rate of vibrations. On this axis, this is how often animals actually vibrate their legs at those frequencies. And then on this axis is showing the action potentials from the two different sensory hairs. And you can see that these are the rates that animals signal at these frequencies, and this really nicely matches the response of each of these two sensory hairs. And it turns out that we actually think that communication by air particle displacement is a lot more widespread. And in fact, we think it's happening in one of the wolf spiders in the genus Schizocosa that we work on in the lab. And one of my students right now, Pallavi Kundu, is testing that hypothesis in Retorsa. Okay, so we learned a little bit about how the legs are used in male-male contests, but again, this study, every time I do anything with Amblipigids, I have many more questions than answers. So I was really intrigued at how they could be moving 30 meters in this tropical, dense tropical forest where I was going out at night and I would often get lost. I had to have flagging every, very short distances so that I could find my own way to my plots and back out of the plots. And these are animals that are, this big, traveling 30 meters. At this point, I pulled in a team of collaborators, Dr. Wolfie Gronenberg, Vern Bigman, and Dan Wegman, and we were interested in testing the hypothesis that aspects of the Amblipigid brain are helping them navigate. And in particular, this is a stain that Wolfie Gronenberg had done where all of this purple area here is all olfactory processing. And then this green are the calyces of the mushroom bodies. So we proposed that Amblipigids are using olfaction to navigate and that they're very sophisticated navigators. And we received funding from the National Science Foundation to pursue this line of research. So the first study that we did, I took advantage of a tropical ecology course that I helped lead through the Organization for Tropical Studies in Costa Rica again. I led it twice, once in 2014 and once in 2017. And in 2014, a whole team of students got together and we tested the role of both olfaction and vision in Amblipigid navigation. So what we did is we collected individuals from the wild, we individually marked them, and then we either covered their eyes to make them blind or covered up their olfactory hairs so that they couldn't smell. And then we released them 10 meters away from their tree, and we would come back night after night to see if they made it home. This was just one study. There were several like this that we did over the years and across a series of studies in the field, some of which even included putting radio transmitters on Amblipigids and looking at where they go. We found that they do indeed navigate using olfaction, and we had some evidence that they might use vision as well. Okay, so just a recap so far. Amblipigids can breathe underwater, they can smell, they can communicate using air particle displacement, and they can find their way home using smell. So the more I read about Amblipigids, the more, again, questions I had, the more fascinated I became. And one of the things that fascinated me most as I started learning more about their peripheral sensory system, their sensory hairs, was their neuroanatomy. And there had been work done by Nick Strausfeld that suggested that the mushroom bodies of Amblipigids, which is a higher order processing center thought to be involved in learning and memory, is huge in amblypigids compared to other animals. And so this is a 3D reconstruction of an amblypigid brain done by Sky Long. And the purple, the two different kind of pink and purple, all of this right here are the lobes of the mushroom bodies. And then the green are the mushroom body calyces. The blue are the optic lobes or optic processing centers. And this kind of magenta at the bottom is the arcuate body. So what I hope you can see is that the mushroom bodies are enormous. They are huge, they are convoluted, and they take up most of the brain. So what is the function? How and why do they have such large mushroom bodies? The first question is, what actually goes into these amazing mushroom bodies? So everything we know about mushroom bodies, for the most part, comes from work on insects. And in insects, the mushroom bodies are sensory integration centers. They're involved in learning and memory. But in the literature, there's a lot of debate as to whether or not the mushroom body of arachnids is homologous or not to the mushroom body of insects. So some incredible work that came out of Wolfie Gronenberg's lab just recently, published in 2020, actually asked this question and looked at what inputs are going into the mushroom bodies. And what they found unequivocally is that both olfactory and visual input are being processed in the amblypigid mushroom body. It is indeed multimodal. So the cyan color here is olfaction, the purple is vision. And what you'll see here in this cartoon is that there is a visual projection neuron that is going into small glomeruli of the mushroom body, and there are olfactory projection neurons that are going into the large glomeruli of the mushroom bodies. So these mushroom bodies are multimodal. They are integrating information from different sensory modalities. Okay, so again, what's the function of these enlarged amblypigid mushroom bodies? So all of the work we had done thus far was done in the field. Everything I showed you was done in the field. We wanted to take a step back and bring them into the laboratory and ask some really specific questions about their cognitive capacity. How well can amblypigids learn? And to do this, and what modalities can they learn? And to do this, we needed a more controlled laboratory setting. So the first study that we did, again, in collaboration with Roger Santer, he led the study, was looking at tactile cues. And it turns out that amblypigids can learn, and they can learn different grains of sandpaper. So how rough or smooth sandpaper is, they can distinguish that, and they can learn to associate one particular type of sandpaper with a hiding place, basically. So the next question was, what about olfaction? Can they learn olfactory cues that are associated with a refuge? So this is a design that has been used a few times, so I'll walk you through it a little bit the first time. Here we have an arena where there's a starting box that the amblypigid starts in and two arms on the arena. On each of these arms, there's a plastic screen that separates a well where you can put a chemical. There are sponges on both sides so that humidity is kept the same. And then we could put different odors in each of these arms. The amblypigid starts in the middle. One of these odors is associated with a crevice that they cannot get in, and the other odor is associated with a crevice that they can get in. The amblypigid is released from the start box. It eventually learns it can't get in that box, but it can get in this one. And the whole time there's a light shining above in order to increase the likelihood that they will be motivated to find a place to hide. So in order to test specifically if olfaction per se is influencing their learning, we also did a treatment where we clipped off the tip of the antenniform leg. And those hairs that can smell are only found at the tip of the leg. So here we have individuals that have their full legs, that have their legs clipped, and then we look at how long they spend with the open crevice. And it turns out that when they can smell, they associate with the open crevice significantly more than you would expect by chance. But when they have their legs clipped, it's actually not different. Then we wanted to ask, how long can they actually remember this? Because memory is also part of the function of mushroom bodies. So we had the experimental treatment here, and then 2 weeks later, we did a retention trial. And it turns out when you do the retention trial, they retain that ability to associate the open crevice with the odor that they had learned. Interestingly, the earlier work I had done with agonistic interactions also showed that Amblipigids could actually remember an interaction with another individual for up to 3 weeks. So they can learn tactile cues, they can learn olfactory cues, they seem to have really good memory. So one of the next questions we wanted to ask is, do they exhibit configural learning? So this is something that is thought to be attributed just to higher order animals. It's thought to be a cognitively challenging task. And basically it's that animals learn multiple cues together in a configuration, and I'll walk you through that. So the question we wanted to ask is, are multisensory inputs processed independently, or are they integrated into a configural representation? So if they are processed independently, if you trained an animal to a visual cue and an olfactory cue, if you paired each of those independently, an olfactory cue and a visual cue, they should remember that and should be able to respond appropriately. However, if they are integrated into a configural representation, if you train them to a combination of a visual and olfactory cue, when you pull those apart, they should not recognize them because they learned it as a configural representation of the two. So this experiment was done at Bowling Green State University, where the cues we used were odors, represented by yellow and blue, and then textures, represented by the darker blue and the lighter blue. So we had these individuals that were trained to just odor or just a tactile cue, and then we had these individuals that were trained to a combination of odor and tactile cue. They went through a training trial where whatever they were trained to, the crevice was open. And then they were tested with odors only, textures only, or a combination of odors and textures. So what we find is that when they were trained on odor and texture alone and then tested, they did really well. They learned the olfaction cue, they learned the texture cue, and they associated with those odors more often than expected by chance. When we trained them to a multimodal cue, they did not respond to an olfaction alone. They did not respond well to a texture alone, but they did retain that memory of the configural representation. So this is consistent with configural learning and is actually pretty exciting that an arachnid can seemingly engage in configural learning. Okay, so we said that the mushroom bodies integrate olfactory and visual cues, so can they learn visual cues? This is another study that was done at Bowling Green State University where they used different patterns of visual representations on both the side of this arena as well as the top. And what they found is that regardless of whether the visual cue was in front or overhead, Amblipigids associated the open crevice more with the pattern that they were trained on. And this is at the beginning of the trial. They ran this training trials over time. And at the end of the trial, they found an even bigger effect of the overhead cues. So they wanted to follow this up and ask whether the median eyes are the ones that are responsible for this potential learning and pattern recognition. So they occluded, excuse me, the median eyes, reran the experiment. Here you have pre-occlusion and post-occlusion, at the beginning of the trials, and then at the end of the session. And you see that before the eyes were occluded, again, they're learning. When they cannot see out of the median eyes, they are not associating with the open crevice. And at the end of the session, after all the trainings, that difference is even larger. So Amblipigids can use olfactory perception in the field to find their way home. They can learn olfactory cues associated with a refuge. They can remember olfactory cues associated with a refuge. They can engage in configural learning, and they can learn visual cues associated with a refuge. And this is better for overhead cues, and they are using their median eyes. Gustavo, you're right, they are so smart. Okay, so next we wanted to say, okay, how far can we push these animals? Can they learn the concept of sameness? And these ideas come from work on hymenopterans, where they continue to show that hymenopterans, bees and ants and wasps, are just incredibly smart animals. So this is the type of design that they use to show conceptual learning, where a bee learns that if it goes through a yellow opening, then the yellow opening is the one that's going to have a reward. And what they find is that when individuals are trained on blue, they show a preference for blue. When they're trained on yellow, they show a preference for yellow after they go through this initial opening. So basically, they're learning that whatever I see here, I need to match that in order to get my reward. So we wanted to see if Amblipigids could do that. So this is research that was done by Fiona Sjogren, who was an undergrad here at UNL, using Phrynis marginimaculatus. Again, the same design that they are using at Bowling Green, where we have a start shelter and then 2 arms with where the odor cues go. What we did here is that in this release shelter, we put the animal in with an odor, and we used either geraniol or 1-hexanol. And then there would be either geraniol or 1-hexanol on one side and water on the other. And the idea was that they would learn that whatever they experienced here would be associated with the open crevice. So we did 10-minute acclimations with the positive odor, 20-minute training trials. We ran 2 trials per day for 5 days. And then we did a test where both sides were closed, and we looked at how long they spent in association with each. We did another 2 days of training trial, and then we used novel odors. So this is where we wanted to see, if you learn this pattern with one set of odors, can you translate that to another set of odors? And that would really show that you're learning the concept itself, sameness. So if they can do this, they should choose the matched side first, whether it's a novel odor or a non-novel odor. And they should spend more time in the matched side for both novel and non-novel tests. So to look at this first one, here we have the training and the novel odor. The incorrect side is going to be in gray, the correct side in red. And what we find for the odors that they were trained on is that they tend to go to the correct side more, but it's not significant. But with the novel odor, we see a significant difference. Their first choice is towards a familiar odor when it's a novel stimulus. So Amblipigids initially choose the match side more often with a novel stimulus. When we look at how long they spend on the matched side, we actually see no difference in the training trial or the novel trial. So they're not spending more time associated with the correct opening, and there were no differences there. Interestingly, okay, so hold on. We see that they matched this prediction for the novel, but not the non-novel test, but didn't match either of our predictions for the time spent. And there's lots of reasons that we think this might be the case. We do think that this suggests learning. Just the fact that we saw this response to a novel stimulus is really promising. But we're not sure that we have the perfect experimental design for this yet. We're not sure that we have the right number of trials per day, the right number of days. And so I feel like we're still it's promising, but we don't have solid evidence that they can learn concepts. They also did not show a difference in time to enter the crevice over the training trials, which again suggests that either we don't have a strong enough incentive or the training maybe wasn't enough. Okay, so Amblipigids can do all these things. They can also potentially learn concepts, and so stay tuned for that. We're going to be retrying some of these experiments to see if we can come up with the perfect assay to really test whether they can learn concepts. So coming back to Peter Weygoldt, he really, in my view, is the grandfather of Amblipigid research. He made an impact on multiple other taxa as well. These are all species that are named after Peter Weygoldt. There are Amblipigids, pseudoscorpions, scorpions, frogs. This alone is a testament to the impact he has made on science. But when you look at the publication records of publications on Amblipigids, and note that his book came out in 2000, and you can see that there is now just a steady increase in publications on Amblipigids over the years. When we look at who the key players are right now, these are the people to watch out for. Michael Sider, Vern Bingman, Jason Dunlap, Kenny Chapin, Gustavo, Dan Wegman, Jonas Wolf, and then a whole series of undergrads that are working with many of these people. So Amblipigid research is on the rise. There are some brilliant, incredible people out there doing some really fascinating work right now, and I can't wait to see what the future holds. And I want to also say that it's not just Amblipigids. Peter Weygoldt also wrote a book on the biology of pseudoscorpions, and that book is having a tremendous impact on one of my students right now, Laura Segura Hernandez, who is doing her PhD on pseudoscorpion behavior. So with that, there are many, many, many people I would like to thank, and I will stop now. Thank you all for coming.